Hello everyone and welcome to Academy. So now as we have done the Prophase 1 of Meiosis 1, we'll move further to other stages of Meiosis 1. But before that, I would like to summarize what all happened in Prophase 1. First of all, the two homologous chromosomes paired together forming synapse. This synapse was accompanied by formation of a synaptonemal complex that is said to stabilize the homologous chromosome during crossing over between the two non-sister chromatids. Now in the diplotin stage, the synaptonemal complex dissolved, but it remained in the portions where crossing over occurred. These portions were referred to as chiasmata. Now in the dikinesis phase, the chiasmata was also finally dissolved or terminalized. This terminalization was in a zipper manner. That means the slipping off of the chiasmata starting from the middle, from where the crossing over started to the end of the chromosomes occurred. Finally, the process of crossing over and recombination is complete. Now also we observe that the centrioles have already doubled during the S phase of interphase. Now the microtubules for the formation of spindle apparatus have also arisen from the centriole during the subsequent stages of Prophase 1. Now as we enter Metaphase 1, the complete formation of spindle fiber has already taken place. Apart from that, the nuclear membrane and the nucleolus has completely disintegrated. So now we start with the Metaphase 1. So this is Metaphase 1, where the spindle formation has already completed and the homologous chromosomes align at the equatorial plate and the two metaphasic plates are formed. Why two metaphasic plates are formed? Here there is a reason. These are the two homologous chromosomes. Each of them has a centromere. Now the metaphasic plate is the plate formation by the centromeres, and we can observe that here two alignments of the centromeres can be observed. Therefore we say two metaphasic plates are formed. In between is the equatorial plate. This plate, equatorial plate, is the plate on which the homologous chromosomes align. Also the nuclear envelope has completely disintegrated as it can be seen that it is completely absent. So this is Metaphase 1. Now the Anaphase 1 is characterized by the two homologous chromosomes moving towards the opposite pole because the spindle fibers are decreasing or reducing in the length. Now there is a very important point. You saw there is one centromere over here and another is over here, but you have seen that no splitting of centromere has occurred in Anaphase 1 as it occurred in case of Anaphase mitosis. No such splitting of centromere occurs in Anaphase 1. But you can observe that the number of chromosomes that move to either sides has become half the initial. Therefore, the number of chromosomes are reduced to half. That is why we call it as meiosis, because the number of chromosomes in each cell would finally reduce to half or lessen up. Finally comes the telophase. Telophase is the terminal phase. Telo means end stage of meiosis 1. Here the spindle fiber completely disintegrates, the nucleus and the nuclear envelope start reforming, and the chromosomes uncoil and elongate. But because the cell still has to complete another phase, that is meiosis 2, these chromosomes are still state. They don't become completely coiled into chromatin material. Now what happens is that only one homologous chromosome out of a pair is received by each cell. If you compare from the start, you saw that a cell have 4 homologous chromosomes. But now as the cell is going to divide, each cell is going to have only two. These are these are two homologous chromosomes, and these are two. The one you are seeing along with the homologous chromosome is the sister chromatid. So you should not confuse it with a chromosome. You can't consider these to be 4. These are only 2 homologous chromosomes. These are 2 homologous chromosomes. So the number has reduced to half in each cell. This can be observed in interkinesis. The cells have separated, and what you see is that only 2 homologous chromosomes are present. The one attached to the homologous chromosome through a centromere is a sister chromatid. So please don't confuse the homologous chromosome with sister chromatids. Now what is interkinesis phase? Interkinesis is a phase between Telophase I and Meiosis II. Before the starting of Meiosis II, there is a phase that occurs, but it is not similar to the interphase, completely similar. Why? Because in this phase, the DNA replication would not occur as in case of interphase, but centrioles replicate. Here the DNA replication is not going to occur. Centrioles will replicate to form 2. Now chromosomes are elongated, but they won't form the chromatid material, as you already learned in the Telophase I, that they won't form the chromatid material. They are elongated, uncoiled, but still they are straight. They won't form chromatin material. Now, though DNA is not replicated, RNA and other proteins which would be required during meiosis II are synthesized during this process. And this finally completes our interkinesis. So basically what we observe is that the initial number of homologous chromosomes was 4. Finally, each cell is going to get only 2 homologous chromosomes. So it is a reductional division. That is why we call meiosis I as a reductional division. Though the basic set of events in case of every stage is same, but there is just a difference between mitosis and meiosis I. The basic difference is that in case of mitosis, the sister chromatids split, but in case of meiosis I, the homologous chromosomes separate. This is important. In case of meiosis II, again sister chromatids are going to split, but that is similar, same as that of mitosis. So there is a basic difference which is between the meiosis I and the mitosis. What is that difference? In meiosis I, the homologous chromosome split. In mitosis, the sister chromatid split in a single go. Thanks for watching. I hope you understand this.